Hello again, this is Melissa Cross. This week we are talking about the muscular system. So this is going to be a presentation on the molecular basis of skeletal muscle contraction. Okay, so we are going to talk about the muscle anatomy before we can get into the molecular basis for contraction, just to help us visualize how this really works. So some things to note. There are three different types of muscles. We have skeletal, cardiac, and smooth. Okay, there are differences between all of them, but for all intents and purposes, we're going to be talking about skeletal muscles today. So, obviously our skeletal muscles are called such because they attach to our skeleton or our bones. They help us move our body, help us pick up things, help us walk, help us talk, all that good stuff. So we're going to be talking about the different subsections of a muscle all the way down to the contractile bit. So the first thing that we're going to talk about is the muscle fiber. So one muscle fiber, so this whole long piece, all these little dots within here are actually all muscle cells. So they usually extend the entire length of the muscle. So this means that they're pretty long, they're pretty skinny. These muscle cells can also then be divided into something called a myofibril. Myofibrils are then differentiated into sections called sarcomeres, and these are the smallest functional units of muscle cells. So this means that they are the smallest section of the muscle cell that can contract and have all the functions of a muscle cell. Muscle cells also have an abundance of mitochondria, and this is going to be important in the following slides when we again talk about the molecular process of this. But basically, mitochondria make a lot of ATP, and that's needed in the process of contraction. Back to these sarcomeres. So this is our smallest functional unit. Within our sarcomeres, we have two very important proteins called actin and myosin. And again, we're going to be talking about those in more detail in the slides to come. The sarcomere is differentiated by something called a Z-line. So the Z-line is just a disc, like two discs on either end, like bookends, and they're going to separate our sarcomeres from each other. Okay, so within that sarcomere, we mentioned two very important proteins. These proteins are called myosin and actin. As you can probably tell from the picture, myosin are considered the thick filaments, and actin are considered the thin filaments. So the actin is this yellow up top portion, and the myosin is the purple portion down at the bottom. We're going to start with actin. So actin is actually surrounded by another two proteins. These two proteins are called tropomyosin, which are these wavy structures here. I'll do that in a different color so you can see it. These wavy structures that intertwine themselves like a braid. And we have troponin, which are these globular-like proteins, which are actually made up of a combination of three amino acids or polypeptides. Okay, so tropomyosin has the job of covering the active sites on the actin so that the muscles are not in constant contraction. This is important because obviously we don't want to have our muscles constantly contract or else we wouldn't be able to use them. Then we have troponin. So troponin actually has a couple different jobs. So when in the relaxed state, when we have tropomyosin over that active site, troponin binds to the tropomyosin, keeping it in place over the active site on the actin. When we want to contract, troponin binds to calcium ions, which are very important for muscle contraction. And once binded to those calcium ions, it unbinds from the tropomyosin, revealing those active sites so that the myosin, these thick filaments down at the bottom, and actin can perform the contraction. And we'll talk more about that in later slides. So now let's talk about myosin, this nice thick purple filament down at the bottom. So as we can tell from the picture, myosin is actually composed of two subunits of the same molecule. So we can tell that based on the two different heads here connected. We have this light purple color and we have this darker purple color. So these two filaments actually twist together and they form like these little golf club like structures here. And they actually have two bend points, and these are very important for the sliding filament theory, which we will go into. So we have a bend point right here where they move away from the whole filament itself, and then we have another bend point right up here at the base of the two heads. And again, this is very important for the contraction of the muscle. On these heads, we have two different binding sites. So we actually have a binding site for actin. So we're going to bind actin to one of our binding sites over here. And then we are also going to have a binding site for ATP, which is made by all those mitochondria, which are very abundant in our muscle cells. So how does all of this begin? Where do we get the signal for a muscle to contract? We know that skeletal muscles are voluntary muscles, which means that we have conscious control over them. So I decide when to pick something up, when to take a step. We have conscious control over them. But the signal comes from something called a neuromuscular junction. So this junction is a chemical synapse, and it's going to relay the information for a muscle to contract by releasing neurotransmitters. In turn, it's going to convert those chemical signals into electrical impulses, which will then trigger the contraction of the muscle. So how does this actually work? In this neuromuscular junction, we have synaptic vesicles, which are going to contain our neurotransmitter. In this case, it's going to be ACH, also known as acetylcholine. This is the main neurotransmitter involved in muscle contractions. Acetylcholine is going to leave the synapse through the synaptic cleft. It's going to pass the synaptic cleft. It's going to bind to the receptors on the postsynaptic sarcoplasmic reticulum, which then will trigger an action potential, and this action potential is going to depolarize the membrane, and that's going to allow for our calcium ions to flow to the myofibrils. So these calcium ions are actually stored up in something called the T-tubules. When the action potential is triggered, the depolarization of the membrane allows those calcium ions to flow to the myofibrils, and they're going to bind on the actin, which is going to cause the tropomyosin to move away from those active sites. The calcium is going to bind to that active site, and then it's going to trigger our muscle contraction. And we're going to put this all together in the next slide. Okay, so we're going to put this all together. So from our neuromuscular junction, we have our acetylcholine releasing our calcium ions to that action potential. Our calcium ions are going to bind to the actin, which is this blue molecule up here in this top animation. So it's right here. Then we are going to also at the same time have an ATP from the abundance of mitochondria, like we talked about before, bind to the bottom of our myosin head. This is going to hydrolyze, which means it's going to break apart, and it's going to form ADP and inorganic phosphate, as shown here by these two molecules, ADP and organic phosphate. The hydrolyzing of this ATP to ADP is going to cause our myosin head to extend up, and it is going to attach to our actin at these binding sites. Remember, our binding sites are now shown because of those calcium ions that attach to our actin and allows the tropomyosin to move away from those active sites. When this myosin head attaches to our actin, this is called a cross bridge. So when this cross bridge occurs, so the binding of the myosin head to the actin, this triggers what is called a power stroke in the myosin head. So at those very important bending sites that we talked about in the myosin, we have this one right here. Sorry, we have this one right here, and we also have this one up here. Okay, this is going to bend drastically, pulling the actin in this direction. Okay, this is where we get our sliding filament theory, because the actin moves past the myosin via that power stroke of that bending action of our myosin head. The myosin is going to pull the actin filament past it and towards the middle of the sarcomere, and this is called the M line. So we talked about those Z lines, the two bookends of our sarcomere, those zigzag lines that kind of differentiate where the sarcomere ends and begins. There's also a line in the middle of our sarcomere called the M line. So this is just, again, M, middle line. The myosin is going to pull that actin towards the M line, and this is going to cause the sarcomere to shorten and therefore contract. So during this, the ADP and the inorganic phosphate are going to detach from the myosin head, and they leave that site open for another ATP to come bind. The myosin head is going to remain attached to the actin until another ATP comes along. The myosin head again will remain stuck there. If a new ATP comes along, again it will be hydrolyzed to perform another contraction, or when that ATP comes along, it will allow the muscles to relax. So there we have it. The molecular basis for muscle contraction just involves some calcium, those filaments, some ATP from the abundance of mitochondria, and boom, we have a muscle contraction.